She is with the School of Biological Science at Victoria. She started her PhD in 2006 at the Malaga Institute of Medical Research. Dianne's supervisors are Dr. Ian Hermans and Professor Franca Wanchis. She is connected with the Zero Puapina mentoring program at VOW. 2 weekends ago, she was trapped in the Rimutaka Forest Park with a group of trampers because of bad weather and had to wait until river levels subsided before getting out safely. So glad to have you here, Dianne.
Good afternoon, everyone. My name is Dianne Sika-Paotonu, and today I'd like to share with you some of the work that I've been doing in designing cancer vaccines. Now, the type of vaccines that I'll be describing to you today are not the sort that you'd be given when you were little, which meant that you would grow up and then never develop disease or cancer. Rather, this is a type of cancer vaccine treatment that would be given to someone who has already developed cancer in an effort to train their own immune system to destroy cancer tissue. Now, the vaccines that I've been working on are dendritic cell based, and dendritic cells are a rare type of immune cell that we all have sitting in our bodies. Now, the problem is that when somebody has cancer, often their dendritic cells are not working properly. Cancers can do things like secrete hormones and chemicals that stop dendritic cells from doing their job properly. Now, the reason that this is dangerous is that the dendritic cells are desperately needed by another immune cell type called a T cell. Now, up here on the screen, you can see a red T cell down the bottom, and you can see a beautiful dendritic cell spinning around. Now, what's important about these red T cells is that they have the potential to turn into cancer-killing T cells. Now, they are unique. We all have them. They live in our lymphoid organs. However, they are very lazy in that they will do absolutely nothing unless they receive the appropriate instructive signals from a dendritic cell that is working properly. Therefore, it was the aim of my project to find a way to make these dendritic cells work harder at converting ordinary lazy T cells into cancer-killing T cells. Now, I chose to go about this using two different methods, the first of which was to incorporate the use of a gram-negative cell wall bacterium, called NPL for short. The other method was to use a sea sponge extract called alpha-galactosaceramide. What we did was we tested these two components separately and then together with our vaccine preparation and checked the type of cancer-killing T cell response that we were able to generate as a result. What we needed to do was, before going into humans, we needed to test this in an animal model, and we used a murine model of skin cancer, or melanoma. And this is what we saw. When we combined our vaccine with the NPL and the sea sponge extract together, we were able to generate a cancer-killing T cell response that was far superior to that which we saw if we gave the vaccine alone. So what these results show and indicate is that it is possible to train an immune system to destroy cancer tissue, and this work forms the basis for an upcoming clinical trial that will be run through the Malaga Institute over the next few years. So thank you for listening. Let's hear it for the dendritic cells and our lazy T cells.
Hi, thanks Dianne again for a lovely talk. It's going to be a hard job for the judges, I think, to sort all these out. I thought that was a very clear presentation. You hit the topic straight away, allowed all the audience to really understand what you're talking about very early on in the talk. I thought you were relaxed, I liked your presentation style, and I think you did a great job.